Hello, fellow microbiomers. My name is Whitney Poulin. Welcome to my presentation. In this presentation, I will be reviewing a study titled Gut Microbiota Restoration Through Fecal Microbiota Transplantation, a New Atopic Dermatitis Therapy. This study was published in Experimental and Molecular Medicine in 2021 by Kim, and looks at the relationship between the gut microbiome and skin conditions. What do we know? Atopic dermatitis is an annoying and painful condition that affects roughly 15 to 30% of children and about 10% of adults. It's likely to first appear during childhood and tends to go away as the child ages, but some people, like me, continue to suffer from it into adulthood. The cause of eczema is not currently known, but someone with a family history of eczema or asthma is more likely to develop the condition, and it tends to be found more in women than in men. Even though we don't know for sure what the cause is, it is thought that disruptions in the skin microbiome is the culprit for this disorder. So why do we care? Right now, there's no known cure for atopic dermatitis. Treatments can range depending on outbreak severity. These range from just applying lotion to taking topical steroid medications. These steroids can have nasty side effects that include thinning of the skin and topical steroid withdrawal. We have seen many studies that have shown a healthy and diverse gut microbiome is important for our overall health. We also know that fecal microbiota transplantation has been used to treat several disorders such as Clostridium difficile infections in the gut, inflammatory bowel disease, and diabetes. If we can understand more about the different disorders linked to gut dysbiosis, maybe we can provide better treatments with fewer side effects. Maybe something like fecal microbiota transplantation is the answer. In this study by Kim et al., they hypothesized that fecal microbiota transplantation might be a more effective treatment compared to probiotics for the treatment of atopic dermatitis, which is also referred to as AD. To start the study, the researchers purchased 30 female mice and randomly divided them into 3 groups with 10 mice in each group. The groups consisted of a donor group, a group the researchers called the Before Fecal Microbiota Transplantation Group, and a Fecal Microbiota Transplantation Group. The mice were kept in a light temperature and humidity-controlled cages with free access to food and water. After the mice were divided, the before FMT group and the FMT group had their dorsal hair shaved, and atopic dermatitis was chemically induced. The before fecal microbiota transplantation group and the fecal microbiota transplantation group both were given antibiotics. The mice were each given an oral dose of one of the 5 antibiotics in one of 2 selected concentrations. Those antibiotics were given in doses of 20 milligrams per milliliter or 100 milligrams per milliliter. The antibiotics given were streptomycin, vancomycin, ampicillin, gentamicin, or a combination of streptomycin and vancomycin. For example, 1 mouse from each group, except the donor group, would receive streptomycin in the concentration of 20 milligrams per milliliter, and a different mouse from each group would receive streptomycin in the concentration of 100 milligrams per milliliter. This was repeated for each antibiotic and concentration combination. Once the antibiotics were administered, the mice were transferred to a new cage with sterilized bedding. The study doesn't state if the mice were housed together or how their food and water were treated after antibiotic administration. After antibiotic administration, the researchers collected fecal matter from the cage of the donor group. The feces was homogenized and suspended in a transfer buffer, and this was given orally to the fecal microbiota transplantation group. This was done a total of 6 times over 2 weeks. After antibiotic administration, the before FMT group was only given the transfer buffer solution without the fecal suspension. Again, this was done a total of 6 times over 2 weeks. The donor group was not given any antibiotics, was not given the chemical treatment to cause atopic dermatitis, and was only administered the transfer buffer without fecal matter. The mice were sacrificed at 8 weeks post-atopic dermatitis induction. In this study, the researchers did several different kinds of analyses. They looked at antibiotic selection and which antibiotics cleared the gut of microbial diversity. They looked at the gut microbiome using 16S rRNA sequencing. They looked at gut metabolite contents by using HPLC to detect short-chain fatty acid contents. They took blood to analyze eosinophil, basophil, and macrophage percentages, and to look at antibody and cytokine levels. They removed the spleens and performed flow cytometry to count and label the different cell types present. They looked at the severity of the atopic dermatitis and did histological examinations to look for mast cells. All of these data were collected and run through different software to perform statistical analyses. Figure 1 in this paper shows the best combination of antibiotic concentration to use for gut microbiome depletion. To get these data, the research administered each antibiotic and its corresponding concentration to one mouse in the before FMT group and one mouse in the group that received the fecal microbiota transplant. They then collected fecal samples, homogenized the sample, plated the slurry, and counted the CFUs, or the colony-forming units. They did this twice, once 24 hours post-antibiotic administration, and once again 48 hours after antibiotics were given. Looking at this figure, part A shows the CFU for each antibiotic at the two concentrations that were administered. The y-axis in part A shows the different antibiotics that were given, with the red bars representing an antibiotic concentration of 100 milligrams per milliliter and the blue bars showing a concentration of 20 milligrams per milliliter. This is plotted against the CFU plate counts and a log base 10. The control sample, which was the feces collected from the donor mice who were not given antibiotics, is plotted at the top of the y-axis, and the other 5 antibiotics are plotted below that. We can see that the 3 best antibiotics for gut microbe depletion is a combination of streptomycin and vancomycin at 100 milligrams per milliliter, this one right here, gentamicin at 100 milligrams per milliliter, and streptomycin at 100 milligrams per milliliter. Streptomycin at 100 milligrams per milliliter is the clear choice for gut microbe depletion, with a 3 log difference between this condition and the control. This equals about a 3,000 CFU difference between the two conditions. Part B of this graph shows the CFU 24 hours and 48 hours post-antibiotic dosing. The graph is set up in a similar way as Part A, with the y-axis showing the antibiotic conditions and the control, and the x-axis showing the fecal CFU and a log base 10. The green bars show the CFU 48 hours after antibiotics were given, and the red bars show the CFU 24 hours after antibiotics were given. This graph shows that it didn't take long for the bacterial counts to start to increase back to the levels of the control. However, it doesn't show the types of bacterial growth going on after antibiotic administration, so we don't know if this is the gut going back to normal microbial diversity, or if this is 1 or 2 bacterial species starting to take over. Figure 2 shows the effect of antibiotics and fecal microbiota transplantation on gut microbiome diversity. These data were generated using 16S rRNA sequencing paired with statistical analysis software. Part A of this graph shows the alpha diversity measure with the observed Chow-1 and Shannon plots. The donor mouse is plotted in blue. The before fecal transplant group, the group that did not receive a fecal transplant but did receive antibiotics, is shown in red. And the fecal transplant group is shown 1 week post-transplant in green and 8 weeks post-transplant in purple. These graphs show that there's a clear drop in diversity after antibiotics are given, with diversity starting to match the control 8 weeks after fecal microbiota transplantation. I don't see at what time point the donor feces and the before fecal microbiota transplant group feces were sampled and analyzed, but I think it would have been interesting to have data showing all three groups at both time points. It would have been interesting to see how long it took for the before FMT group gut microbiome to shift back to that of the control, or if this shift would even happen at all. Part B of this figure shows a PCOA plot where the colors of the dots mimic the colors of the alpha diversity measure. So blue is the donor, red is the no fecal transplant, green and purple are the fecal transplant group 1 week and 8 weeks post-transplant, respectively. The researchers call out that the gut microbiome communities were clearly divided between the no fecal transplant and the fecal transplant group, but I don't see it. I don't have much experience interpreting these plots, but I don't see a clear divide. The red dots do cluster more at the bottom left of the graph, with the green and purple shifting to the upper right, but I'm not convinced that this is a clear divide. So what do you guys think? Part C of this figure shows the relative abundance of bacteria in the gut for each condition. The plot on the left shows the bacterial phylum found. The graph in the middle shows the bacterial family, and the plot on the right shows each genus found. Each plot has the donor group on the far left of the x-axis, followed by the before FMT group, the fecal group 1 week after transplant, and the fecal transplant group 8 weeks after transplant. These plots do clearly show a significant drop in bacterial abundance, with the abundance starting to be restored 1 week after the transplant occurred, and the abundance fully restored 8 weeks after transplantation. It looks like the biggest hit might have been on Lactobacillus, but that might just be because that is the genus with the largest abundance. It would be interesting to see these plots paired with a table of percentages outlining the loss of abundance for each group. I like the visual, but I think the numbers would also help us tell more of the story. Figure 3 shows the levels of short-chain fatty acids present in the gut after antibiotics were given and 8 weeks after a fecal transplant. These data were generated using HPLC. In each plot, the fatty acid is plotted on the y-axis and the experimental conditions are plotted on the x-axis, with the donor group plotted on the left, the before FMT group in the middle, and the fecal transplant group 8 weeks after the transplant plotted on the right. Plot A shows acetic acid levels. We see a significant drop of about 200 milligram per liter of acetic acid present in the blood after antibiotics were given. This level is shown to increase 8 weeks after fecal transplantation to match the level seen in the donor group. Plot B shows butyric acid. We also see a significant drop in butyric acid levels, but these levels are not restored quickly like the acetic acid levels were in the 8-week post-FMT conditions. These levels appear to only increase by about half of what was lost. Plot C on the lower left shows isobutyric acid levels in the blood. Again, we see a significant drop in levels after antibiotics were given, with about half of the loss restored after FMT. The last plot, plot D, on the lower right shows propionic acid. This also saw a decrease after antibiotic use with an increase after FMT. Figure 4 is a heat map showing cytokine concentration in the blood serum. These data were generated using enzyme-linked immunosorbent assay kits. Each column of this map shows the concentration of cytokines for each individual mouse, with the 10 donor mice clustered together on the left, the 10 before FMT clustered together in the middle, and the last 10 columns representing the FMT group 8 weeks after receiving a fecal transplant. The top two rows show the cytokines secreted by T regulatory cells. The row in the middle shows cytokines associated with Th2 cells, and the three rows on the bottom are cytokines associated with Th1 cells. The different colors show the concentration of cytokines found, with green being the lowest and red being the highest concentration. As you can see, the donor mice start out with higher levels of cytokines from Th2 cells and TNF alpha from Th1 cells. All of the other cytokines measured are about the same concentration. As antibiotics are given, there is an increase in cytokine secretion from T regulatory cells and Th2 cells, and a decrease in TNF alpha concentration. These concentrations start to decrease after fecal transplant in the FMT 8-week group, but the concentrations still don't match the donor group. Figure 5 shows the effect of fecal microbiota transplantation on T cell activation. These data were generated using flow cytometry. These plots show CD86 and CD274. CD86, or cluster of differentiation 86, is a protein that is found on the cell surface of antigen-presenting cells. These cells are needed for the activation of T cells. Part A of this graph shows CD86. I'm not too familiar with interpreting graphs like these, so I'm not sure of the units that it's portraying, but you do see an increase in the mean fluorescence intensity after antibiotics in the before FMT group, with that decreasing 8 weeks after fecal microbiota transplantation. I'm assuming that this means that there are more CD86 cells present after antibiotics are given, so more T cells are being activated. Then T cell activation would start to decrease as the microbiome is restored through the fecal microbiota transplantation. Part B of this figure shows CD274. CD274 is also called PDL1, or programmed death ligand 1. It is found on activated T cells, and it signals other cells to go through programmed cell death. In these plots, we see that there is a decrease of CD274 after antibiotic administration, with an increase to slightly past the donor population after fecal microbiota transplantation. This suggests that there are fewer T cells signaling apoptosis after antibiotic use, and FMT restores these cell populations back to normal. Figure 6 shows the effects of antibiotics and fecal microbiota transplantation on antibodies and immune cells in the blood. These data were generated by serum cell counts. Part A shows the IgE concentration in nanograms per milliliter on the y-axis, with the donor group, the before FMT group, and the FMT 8-week group plotted from left to right on the x-axis. We can see that there is an increase in IgE antibody concentration after antibiotics are given, with those concentrations returning to almost match the donor concentration 8 weeks after fecal transplantation occurs. Part B and C show the eosinophil percentage and the count per microliter respectively. These plots are the same as A, where there is an increase in count and percentage, with those concentrations decreasing back to match the donor concentration after fecal microbiota transplantation. Plot D shows the basoeosinophil percentage, plot E shows the monocyte percentage, and plot F shows the calprotectin concentration in picograms per milliliter. These three plots all match the other three plots, which show a significant increase in concentrations or percentage and a decrease after fecal microbiota transplantation that goes to match the donor concentrations. Figure 7 shows the effect of a fecal microbiota transplantation on the skin and the gut cell composition. Part A shows an image of a mouse dorsal skin after the gut microbiome was depleted and atopic dermatitis was chemically induced. The image on the left is the group that did not receive the fecal transplantation, and the image on the right in part A is the mouse that did receive a fecal transplantation. The bar graph to the right of those images show the dermatitis scores that were given to each mouse. The donor group showed no redness or irritation. The before fecal microbiota transplantation group showed significant redness and irritation after atopic dermatitis was induced and antibiotics were given. And the fecal microbiota transplantation recipient group showed a significant increase in redness and irritation after antibiotics and atopic dermatitis was induced, with that redness and irritation significantly decreasing after a fecal transplant. Parts B and C show images from stained dorsal skin and stained ileum, respectively. The red arrows that overlap the pictures are pointing to the mast cells in each image. In the before FMT group in Part B, you can see a significant increase in the number of mast cells after atopic dermatitis was induced and antibiotics were given. These mast cells decreased significantly in the FMT group, as shown in the image to the right, 8 weeks post-transplantation. I'm not sure that the ileum images were the best ones to use because it doesn't show a significant difference in mast cells, even though it is indicated that there is a significant difference in mast cells present in the bar graph to the right of those images. The last figure in this paper, figure 8, is a cartoon showing the mechanism effect of fecal microbiota transplantation on atopic dermatitis. This image just wraps everything up into one cute little cartoon. Starting on the left, we have a mouse with atopic dermatitis being chemically induced. After that, we see a decrease in the concentration of short-chain fatty acids in the gut. We also see a decrease in cytokines signaling the Th1 cells, with an increase in T regulatory and Th2 cytokines, which results in an increase of concentration of these immune cells. And we also see an increase in antibody production. This antibody increase results in an increase in itching, which results in an increase in redness and inflammation on the dorsal skin of the mouse. If we move over to the right side of the graph, this mouse would have received the fecal transplant from the donor. After fecal transplantation, we see an increase in short-chain fatty acids. We see an increase in the cytokines that are signaling the Th1 cells. We see a decrease in the T regulatory and Th2 cytokines, a decrease in our mast cells, and a decrease in antibodies. This results in a decrease of itching and a decrease in redness and inflammation on the dorsal skin of the mouse. In this study, the researchers are trying to link probiotic use and fecal microbiota transplantation. They're stating that FMT is a better option of the two. The researchers cite that a few studies have shown gut microbiome improvement with probiotic use, while other studies have shown no improvement, and state that FMT might be a more useful approach. In this study, there was an increase in microbiome alpha diversity with a high abundance of Porphyromonadacea, Lactobacillia, and Rickenellacea. Those are hard, y'all. After FMT. These phyla of bacteria are suggested to play an important role in immune response and the restoration of a healthy gut microbiome. Some of the study challenges that I noticed with this study were the researchers didn't use both male and female mice. That might have been because eczema is prevalent mostly in human women, but I think it would have been a good idea to include both male and female mice. The researchers wanted to draw a line between probiotic use and FMT, but they didn't include a probiotic-only condition in their study. In their discussion, they compare probiotic use a lot with their results from this FMT study, but I don't think it's an accurate comparison since that condition was not included here. The last challenge I noticed was that the sample size for each condition was pretty small. To conclude, this study showed that with gut microbiome restoration studies like this, we can begin to understand how our microbiome impacts our immune responses. With this study, we learned that the skin microbiome may not be the only thing to blame for skin conditions. Our gut microbiome has an impact on our skin health as well. To get our discussion going, here are my questions. So 1. What are your thoughts on fecal microbiota transplantation? If you were a patient in need, would you hesitate or would you opt for the treatment right away? 2, if you could talk with the researchers about this study and their findings, what comments would you make and what questions would you ask? 3, these researchers also looked at restoring the gut microbiome with cream cheese. What other things would you suggest for them to try in their next study? 4, what do you think about the PCOA plot in figure 2 that I talked about? Do you see a clearly divided group, or do you, like me, see them all clustered together? And 5, what challenges did you see with this study? Here is my list of references that I have referenced throughout this talk. And finally, thank you so much for listening to my presentation. I'm really looking forward to our discussion about this paper. Bye, y'all.